Our today's topic is oxygen transport. The objectives are: explain the oxygen transport mechanism, significance of hemoglobin oxygen curve, Bohr's effect, and those factors which shift the oxyhemoglobin curve. OHC stands for oxyhemoglobin curve to the right or left. Now, first of all, you have to remember that typical partial pressures of oxygen and carbon dioxide in atmosphere and in various sites in the body. Now, first let us take oxygen. That in the atmosphere, the partial pressure of oxygen is 160 mmHg. But in the alveoli of the lungs, it is about 100 mmHg. Now, this 100 mmHg is present in the alveoli, where the pulmonary vessels, they exchange the blood. In the pulmonary capillaries, the blood comes to the alveoli, and here the exchange takes place. So just remember, right, that the partial pressure of the oxygen in the alveoli is 100. In the pulmonary veins, it is 100. In the systemic arteries, it is also 100. But I would say that it is a little lower in the systemic arteries. Then there in the cells, it is about 40. So if you see over here, right, that there is a gradient for diffusion of oxygen from the alveoli to the lungs, and the systemic veins which bring the blood for oxygen towards the alveoli. So please remember these partial pressures. Now, at the lung level, what happens? As I already told you, that partial pressure of oxygen in the alveoli is 100 mmHg. Now, the blood that is coming for oxygenation to the lungs, now remember, it comes through the pulmonary capillaries. Now, if you see over here that this is a end of the pulmonary capillary, which is called as the arterial end of the pulmonary capillary, and this is called as the venous end of the pulmonary capillary. Remember that this is the arterial end of the pulmonary capillary, but it is having the venous blood, and this is the venous end of the pulmonary capillary, but it is containing the arterial blood. Now, if you see that the blood from the systemic veins is coming to the pulmonary capillary with the partial pressure of 40 mmHg. Now, when this blood comes to the alveoli, in the alveoli the pressure is 100 mmHg. So there is a diffusion gradient of 60 mmHg. So when the blood passes from this pulmonary capillary, it becomes in equilibrium. In equilibrium means that this 40 mmHg partial pressure of the oxygen which is coming for oxygenation to the lungs becomes 100. So the blood that comes out from the pulmonary capillaries is 100 mmHg partial pressure. This is the point you have to remember. Now, this is the pulmonary respiratory membrane, this is the alveoli, these are the alveolar cells, and this is the capillary. This is the capillary endothelium, this is the alveolar epithelium. Oxygen has to pass this structure. This is the basement membrane of the alveoli, this is the basement membrane of the pulmonary capillary. So oxygen has to cross all these structured respiratory membrane and enter into the RBC for combination with the hemoglobin to form a compound oxyhemoglobin. Now what happens at the tissue level? The blood that is coming to the cells or the tissues, it is having a partial pressure of oxygen at 100 mmHg, and the cells have the partial pressure of oxygen of 40. So definitely here the gradient is favor for the pulmonary capillary blood to diffuse into the cells, and the blood that moves out of the pulmonary capillary, this has got the partial pressure of the oxygen. So the arterial blood that comes with the partial pressure of 100, but when it goes to the veins, the partial pressure becomes 40. This happens at the tissue. Now oxygen is carried in the blood in two forms. It is bound to hemoglobin as oxyhemoglobin. Hemoglobin is present in the red blood cells, or it might be dissolved in plasma, normally insignificant because oxygen is not water soluble, so it has got a very low solubility coefficient and it does not dissolve in the plasma water. So a very small amount of the oxygen is present in the dissolved form in the plasma. Main is that it is carried in the blood in combination with the hemoglobin as a compound oxyhemoglobin. Now each heme molecule, as you know, is capable of binding with one oxygen molecule, and each globin molecule is capable of binding one carbon dioxide molecule. So remember, there are 4 iron atoms in each heme molecule, and each iron atom can combine with one molecule of oxygen or 2 oxygen atoms. Now each molecule of hemoglobin can bind to either 4 molecules of oxygen and 1 molecule of the carbon dioxide. So 100 ml of blood has about 15 grams of hemoglobin normally, when the hematocrit is 0.45 or PCV is 0.45. Now this is a mechanism which is called as the heme-heme interaction. Here if you see that one hemoglobin molecule will combine with oxygen, it will form oxyhemoglobin. This will facilitate the combination of the other oxygen, the third oxygen and the fourth oxygen molecule. So this shows that combination of one molecule oxygen with each one iron will facilitate the combination of the second oxygen molecule with the second iron atom in the hemoglobin, likewise the third, likewise the fourth. This is called as the heme-heme interaction. Now oxygen transport at normal pH in blood with hemoglobin. Again, you recall this is the alveoli, the partial pressure is 100, but the blood that is coming to the alveoli for the oxygen, right, there it is equilibrated with the partial pressure of 100. So this blood which leaves the pulmonary capillary, it is 100 mmHg partial pressure of the oxygen. But when it comes into the systemic artery, due to the mixture of some blood that is not oxygenated, and this is due to the structural formation, some blood is mixed with the venous blood, and therefore in the arteries this pressure of oxygen, partial pressure of oxygen, it drops to 98 or 99. And in some cases, the hemoglobin is also not pure. So red blood cells with hemoglobin are carrying 98% of their maximum load of the oxygen. So in this way, the blood which passes from here to the pulmonary capillary, it has the partial pressure of the oxygen 100 ml. But when it goes to the left side of the heart, there is some venous blood which has bypassed the oxygenation like mixed, and this reduces to 98 or 99. So the O2 content of the plasma is equal to 3 ml oxygen per liter of the blood, right? Now if you see, as I told you, that is a very small amount, about 3 ml oxygen per liter of the blood, very small amount. But O2 content of the red blood cell is 197 ml O2 per liter of the blood. So total oxygen carrying capacity is about 200 ml oxygen per liter of the blood. Oxygen transport in the blood without hemoglobin. Now, let us think that if the hemoglobin is not there, alveolar PO2 is equal to arterial PO2. So, alveolar PO2 is 100. So, arterial plasma, there will be exchange taking place. Partial pressure of the oxygen is 100 mg. So, O2 content of the plasma is 3 ml oxygen per liter of the blood. But O2 content of the red blood cells is 0. So therefore, total oxygen carrying capacity when the hemoglobin is not there will be only 3 ml O2 per liter of the blood. Now this is a typical oxyhemoglobin dissociation curve. Right. Now this central curve is the normal position, but during respiratory cycle this curve shifts to the right or shifts to the left. So try to understand that what does it mean. Shifting of the curve to the right means that there is a decreased affinity between the hemoglobin and oxygen, or you can say that the oxygen can leave the hemoglobin easily. But when this curve shifts to the left, there is an increased affinity for the oxygen with the hemoglobin. So there is a very rapid binding. When the curve shifts to the left, there is a rapid binding between the oxygen and the hemoglobin. When the curve shifts to the right, there is a decreased binding and easy release of the oxygen. You can now understand that when there is increased affinity, here the partial pressure of oxygen is 40. So the hemoglobin saturation comes to about 80 at 40 pressure because of the increased affinity. But when the affinity is decreased, you need more pressure to get the 80% saturation. So this means when you require more pressure for the binding or saturation, this means the affinity is decreased, or in other words you can say that the oxygen would be easily released from the hemoglobin. But in case of the leftward shift, this is opposite, that small pressure would give more saturation and there will be a strong binding between the hemoglobin and the oxygen. These points are very important. Try to understand them and try to remember them in the oxyhemoglobin dissociation curve. And you might surely get a theory question from this part of oxyhemoglobin dissociation curve. Now there is a summary. What factors can shift the curve to the right or the left? So these are the factors, right, which can shift the curve to the right side. That is the decrease in pH, number one, increase in hydrogen ion concentration, number two, right. So what they will do, that they will shift the curve to the right side. Shifting of the curve to the right side means again, right, that the affinity between the oxygen and hemoglobin will be weak and oxygen will be easily released from the hemoglobin. Increase carbon dioxide, right. So the effect of the pH or effect of the carbon dioxide, increased carbon dioxide would also decrease the affinity between the oxygen and the hemoglobin. Increase in temperature would also decrease the affinity or shift the curve to the right side. Increase in 2,3-DPG. This will also reduce the affinity or shift the curve. So whenever the affinity is reduced, it will shift the curve to the right side. DPG, 2,3-DPG is increased in cases of the hypoxia, in cases of the exercise. So if there are other conditions where the 2,3-DPG is increased, acclimatization to high altitudes, chronic lung disease, anemia, hyperthyroidism, right to left shunt, congenital heart disease, pulmonary vascular disease. So shifting of the curve to the right will always occur in those conditions when there is increased cellular demand of oxygen, increased metabolism. Due to the metabolism, temperature is increased, there is more production of the hydrogen ion, there is a decrease in the pH, there is more carbon dioxide, there is more 2,3-DPG. And reverse all these conditions for the shifting of the curve to the left side. But to this add fetal hemoglobin or HeF. So all those conditions opposite to the shifting of the curve to the right will be shifting the curve to the left.